Now that you're familiar with what a phylogenetic tree is, we want to talk about the data that's used to construct these trees. Fossils are one type of data that can be used, and another type of data would be morphology. Now, morphological data is used because when organisms are closely related to each other, they do tend to resemble each other. Now, this will not always be the case, but commonly this does work. So if we look at these different appendages here, we've got man, cow, horse, whale, and bird. And we can see that for the man, this is an arm that we're talking about. We've got a leg for the cow and the horse. We've got a fin for whale, and we've got a wing for bird. But regardless of the fact that these are very different organisms, when we look at this appendage, the bones do tend to match each other. So if you just look at the color patterns here, these are going to be what we would call homologous structures. And these are homologous because they're similar to each other because they came from a common ancestor. So we're going to say that all of these organisms are similar because there was this common ancestor way back that had these different bones in their appendage. And then these evolved from that common ancestor, and they were affected by different selection pressures. And then that led to those appendages being a little bit different, where you see some longer bones in one, some shorter bones in another. Now, it is possible for organisms that are closely related to look very different as well. And this is why it does become important for scientists to use a range of different data sources when they're trying to construct these phylogenetic trees. If we look at this little comic here, we've got the two girls who are similar due to inheritance. So if we assume that these are twins or that these are sisters, notice that they do look alike. They don't look exactly alike, but we do see some similarities there. So we have a similar hair color, we've got similar skin color. You could say that they're dressed in a similar manner. This would be what we would call homologous. Again, homologous refers to the fact that they have a common ancestor. So this is something that is related to their inheritance. They come from the same parents, so they're going to look alike. That's the reason for it. Over on the other side, we have two individuals that are similar due to other factors. So they look similar to each other because they decided to dress up so that they look similar to each other. This is going to be what we would call analogous. This is analogous because this is not due to inheritance, it's due to something else. And when we're talking about morphology and using that to construct phylogenetic trees, when we see organisms that look similar to each other, the question that has to be asked is, do they look similar because they inherited those similar characteristics from a common ancestor, or do they look similar just because perhaps they were subjected to similar selection pressures and they happen to evolve the same types of appearances? Here's just another example of yet another analogous structure. We have basically a sugar glider, and then we have a flying squirrel. These occupy similar habitats or similar niches. They both live in trees, and they use these wing flaps or these flaps of skin that help them to glide through the air. So they're not really flying, they're just gliding. These are not going to be homologous structures because the most common ancestor that we can find for these two happens to be this rat-like organism down here. And here we have no skin flaps. So if we have no skin flaps in that most common ancestor that we have for these two organisms, we have to assume that those similar structures evolved independently. That makes them analogous to each other. They're not due to being inherited from the same ancestor. Another tool that we can use for constructing phylogenies is molecules. And when we're talking about molecules, a lot of the time we're talking about DNA. This is certainly not the only molecule that we can use. We could use mitochondrial DNA, which is often the case. We could use nuclear DNA. We could use DNA for ribosomal genes. We could also look at proteins. That would be another thing to compare. But when we do this, first step would be to sequence the organisms and get the DNA, and then we can look at how that DNA might have changed over time. So we can compare the DNA and see whether we have exactly the same sequences there, the same nucleotides there, or whether there's been insertions or deletions. And then we can try to make some kind of educated guess as to how two organisms might be related to each other. So when we use molecules like DNA to trace phylogeny, the first thing we have to do is we have to obtain that sequence information. So there would be some sequencing involved in the process. We would have to obtain DNA sequence from multiple organisms. We would try to obtain some sequence information from what we think to be the common ancestors as well. And then we would align or compare those with each other, and we would try to use them just in the same manner as we would a morphological character. So we would be looking for similarities and differences between the organisms, and then we would also compare that to what we would guess to be the common ancestor of the two. If we look here at an example of this, this is a protein sequence that is aligned among 5 different species. And this shows you how complicated it can sometimes get, because you're not going to have necessarily the same number of amino acids to be compared. Some might have insertions, some might have deletions. And so there's special software that's going to help to align the sequences with each other so that we can get a better idea of how similar or how different they happen to be. Notice that in some cases it's necessary to introduce just some gaps in one of the sequences because maybe another one, such as the second one in this list, has some extra amino acids there. The black areas on this show where they are similar or where they are the same as each other. And then where you have the white is indicating that there's quite a bit of difference there. So right here, this would be a region where we have high similarity. And then when we look at something like this, this is low similarity. If we look at another example, this is showing us a DNA sequence that's aligned, again, among 5 different species. And you can see how some of the sequences have some gaps introduced there because there have been some insertions in some of the sequences. And we can also see that there are varying levels of black shading throughout this. And the black indicates to us that they have the same sequence, so those would be places where they're matching up quite well. It can be difficult to do these types of sequence alignments by eye. So again, there's software that does help to compare sequences with each other, to generate alignments such as what you see here, and come up with essentially some percentages that say how similar the genes are to one another or how different they are to one another. We can have what's called orthologous genes, and then we can also have what are called paralogous genes. And so when we're talking about orthologous genes, these are genes that are due to basically speciation. What this means is that there was an ancestral gene, so that common ancestor had this gene, and then over time, due to different selection pressures, this gene became a little bit different in 2 different species. So that when we look at this gene in these 2 species, it is different. It may have a different role altogether in the 2 species, or it may be very similar to each other. But it's one gene that once was the same single gene in that common ancestor. That's the orthologous genes. When we talk about the paralogous genes, these are genes where basically they have been duplicated over time. So we have one ancestral gene, and that ancestral gene has been copied a number of times. This could be due to an error somewhere along the way in DNA replication or in the cell cycle process, but somehow we have multiple copies of it. Once you have multiple copies, once you have that gene duplication event take place, then these genes may begin to take on different roles in that organism. So one gene, if we call this gene number 1, gene number 1 may continue to do what the ancestral gene did. Gene number 2 may take on a new role. Either way, we have 2 different genes now in one organism that both came from that one ancestral gene. That's what we mean by paralogous genes. We can also look at DNA sequence, and we can compare the entire sequence from one organism to multiple other organisms. And then we can talk about how similar they are just sequence-wise. If we talk about humans, their DNA sequence is 98% similar to a chimpanzee's sequence. 44% similar to a fruit fly, 26% similar to yeast, and then we've got it 18% similar to plants. So that's the percentage of our genes that we actually share with these other organisms. We can also look at how many genes are present in an organism. There isn't a real good relationship between complexity, or what we view as complexity of an organism, and the number of genes that they have. Humans have a very similar number of genes to what we would find in a mouse. So we're looking at maybe somewhere around 30 to 40,000 genes present in those. We could talk about other eukaryotic cells. We like to think of ourselves as being quite more complicated than a nematode, but C. elegans, the worm you see on all of this, it has close to 20,000 genes. So it has over half the number of genes that we have in humans. You can also see that we've got some plants rather high on this as well that have numbers that are approaching humans as well. It is common for eukaryotic cells to have quite a few more genes than what you would find in prokaryotic organisms. So as we talk about all of this molecular data, there are a number of advantages to using this type of data. One of them is that there is an equivalence there. A nucleotide is a nucleotide regardless of whether we're talking about a bacterial cell, a human cell, a plant cell, a fish. It is equivalent to each other. Same thing if we're talking about a protein. Amino acids are amino acids no matter what we're looking at. That is a big advantage here. Another thing is that we have an enormous data set. When we're talking about humans, we have 3.9 billion nucleotides in our DNA. Other eukaryotic organisms are going to have billions or millions, most of the time, nucleotides when we compare them. So there's a lot of data to work with there, and then that makes statistical analysis possible as well. Another thing is that if you have a molecule that changes very rarely, then we can use that to trace relationships back a very long ways in evolutionary history. That is very useful. We can also avoid all non-heritable variation. When we just look at the phenotype of an organism, phenotype can be affected by heritable things. It can also be affected by environmental things. And when we're just looking at an organism, it can be difficult to rule out all of those things that are due to the environment. If we're looking at the hereditary molecules themselves, we don't have to worry about that non-heritable variation influencing any decisions that we're making here. This doesn't mean that it is all advantages when we're talking about molecular things being used for determining phylogenies. There's certainly some disadvantages there as well. And one of those is that there's no record of the past changes. So you could have changes that happen, and then you could have another change that happens, and we never are able to see that first change. So we're not getting the record of all of the changes taking place. Keep in mind there's only 4 nucleotides. We have A, T, C, and G. And so 4 real changes are possible there. We can't tell if it's changed multiple times. So there's no recognizable intermediate condition between the characters. We see the change that was in the past at the one moment that we looked at it, and we see the change that's now in this organism that we're looking at. We see nothing in between. So when we're talking about molecular clocks, And by molecular clock, that is a gene or a DNA sequence that we're looking at to determine evolutionary relationships. These are going to calculate time that's elapsed based on DNA sequence differences. In this case, we're assuming that we have a gene that's going to evolve or change at a constant rate. This is an assumption. It's not always going to hold true, but most of the time, scientists are able to pick out genes that do seem to change relatively constantly. We're then assuming that the difference between the sequences of DNA is proportional to the time since the species evolved from a common ancestor. So what we're able to do with all of this information is help to draw up that phylogenetic tree and then also get some rough idea of evolutionary time. So this means that we can try to put some type of a numerical value on the number of years that have evolved from that common ancestor to the organisms that we're looking at today. Here's what we're talking about with this molecular clock. We can see here we've got a common ancestor. We can look at the changes that have taken place, and even though these two sequences that we're looking at here in this middle box, they have a different change, notice they have one change. For this gene, we get one change every 25 million years approximately. So if we look at a sequence and we see that we have one nucleotide that has changed, we can assume that it has been approximately 25 million years. If we look at a sequence and we see two nucleotides have changed, then we can make the assumption that it's double that. So we can say 50 million years have passed. So in this way, we can get a rough idea of geological time with these sequence changes. On this graph, we can see that we have number of mutations versus millions of years, so divergent time. And what we can see is that as more time has passed, we have more mutations take place within a gene. If we're talking about a gene that is a good one to use as a molecular clock, then we get this nice linear increase in the number of mutations when we do plot these values, number of mutations versus the divergent time.